We will spend the next few minutes discussing various aspects related to large-scale chromosomal changes. For example, we'll talk about chromosome deletions, duplications, and various rearrangements, including inversions and translocations. In addition, we'll talk about changes that occur regarding changes in chromosome number within cells. Let's just begin by making the point that chromosomes, although they're stable, they can break. And changes can occur not only between homologous chromosomes, but also between non-homologous chromosomes, as we see illustrated in the image on the screen. So chromosomes, they can break, and when they do, they can often recombine. In addition, chromosomes can cross over between repetitive DNA sequences. So there are various ways by which changes in chromosome structure can occur either by breaking and rejoining or by crossing over between repetitive DNA sequences. This is a figure that illustrates some of the changes that can occur regarding the structure of chromosomes. So if we begin in the middle, where we see a base chromosome or a wild type sequence of portions along a chromosome, so red, green, brown, and blue, again just as a way to schematically represent a sequence of regions along a chromosome. What are some of the changes that can occur? Well, over here toward the top right in yellow, we can have a loss of genetic material. For example, we can have a deletion of a gene or of multiple genes, or we can have some mistakes that result in the omission of an entire chromosome. In addition, toward the lower right in dark blue, we can have various mechanisms by which we have a gain of genetic material. So for example, we can have duplication of genes. That's what we see illustrated by this chromosome with two brown regions now. And we can actually form duplications of entire chromosomes or even entire sets of chromosomes. Beyond that, toward the far left, we have changes that can occur regarding reallocation or a shifting of genetic material. So on one hand, toward the bottom left, we have an inversion. We have all of the same portions of our base chromosome, but notice we've just flipped the orientation of the brown and the green regions. Another possibility is moving large sections of chromosomes between non-homologous chromosomes. In other words, it's referred to as a translocation, and that's where it's illustrated in this upper left-hand chromosome. So this region right here in pink came from a totally different chromosome. So again, this just illustrates some of the changes that can occur regarding the structure of chromosomes. And just a few points to keep in mind. Typically, if you have major mutations or changes in chromosome structure, often those chromosomes are lost, those cells simply die, so we typically just wouldn't see those very often. That's not always the case. Certainly we can have some chromosome changes that persist and can often cause problems. For example, we'll take a look at one case that causes lymphoma. And also, some of these chromosome rearrangements play a big role in evolution and speciation. We'll talk briefly about that in just a bit. One example of, so let's just spend the next few minutes discussing sort of not the details behind how we get deletions, duplications, inversions, and translocations, but let's just talk briefly about some of the implications behind these changes that occur. So one example of a syndrome that is genetically based is Williams syndrome. This is a situation, it's found in many people, and it generally causes them to be sort of gregarious, very socially active. They love music, and often they have perfect pitch. But they also have some health defects as well, problems with their heart and so forth. So what is the genetic basis behind Williams syndrome? Well, it's illustrated in this figure here. We're looking at normal sequence of part of a gene, of part of a chromosome, and illustrated toward the top, just briefly, we have these two regions here labeled PMS. So the PMS is a gene, so there are two copies of this gene. Between the PMS gene, there are 17 other genes. And what we can have imagined during meiosis, recall crossing over, that occurs during prophase 1. If we have crossing over between two chromosomes, and if those chromosomes aren't exactly lined up correctly like we see here, again, we can have crossing over between repetitive DNA sequences. So that's what's illustrated here. If that happens, we end up with one chromosome that lacks the region in blue and one chromosome that has a duplication of all those regions in blue. So again, that's just these 17 genes. And the problem is the chromosome that lacks those 17 genes, if that happens to end up in a sperm or egg cell that fertilizes another gamete that is, let's say, normal, that individual will have only one copy of these 17 genes, and as a result, of course, it's not a lethal condition, but as a result, that will cause that person to end up having Williams syndrome. If we think about chromosome deletions for a few more seconds, we have a few examples illustrated here. On one hand, we can lose portions located on the end of chromosomes. So the blue F indicates a region that will end up being deleted. We'd say, well, that's an example of a terminal deletion. On the other hand, we can lose regions not on the end of chromosomes. So we'd say, well, that's an example of an interstitial deletion. So in addition to deleting or losing parts of chromosomes, we can also have chromosome duplications, where we duplicate or form extra copies of parts of chromosomes. And that's illustrated on the left. So we have this region here indicated by D and E duplicated. So now we have 2 copies of D, 2 copies of E. Similar situation on the right, but now we have the situation in both chromosomes. So what this can result in, if you think about it, is a change in gene dosage. So now you have 2 or more copies of various genes, whereas you originally just had perhaps just 1 copy of each of those genes. And what can happen over time is that these extra copies that are produced can play a big role in evolutionary divergence. So in other words, now you have 2 copies of various genes. Perhaps there's no real need to have both of those genes doing what they were originally intended to do. So now they can accumulate mutations over time and take on different roles as populations and species evolve. Another example of changes in chromosomes that can occur are inversions. So all we're talking about here is simply changing the orientation of parts of chromosomes. We're not losing any DNA, we're not losing any part of the chromosomes. All we're doing is just simply rearranging the order in which various genes are located along a chromosome. So we have a couple of examples here. So toward the top we have just, if you notice the region in blue, this region has been flipped from what we have on the left-hand side. And so on one hand we can talk about inversions that occur on either side of the centromere, and those represent examples of para-centric inversions. We can also have inversions that span across a centromere. So we say those are examples of peri or pericentric inversions. Another example of changes that can occur along chromosomes are translocations. And translocations are actually movement of large pieces of chromosomes between non-homologous chromosomes. So this can have a variety of implications. For example, one type of lymphoma, Burkitt lymphoma, is caused by a translocation between 2 different chromosomes in humans. So that's illustrated here. We have 2 chromosomes illustrated, so chromosome 8 in red, chromosome 14 in yellow. And I just want to point out that along chromosome 8, it of course contains lots of genes, but one of the genes on chromosome 8 is this MYC gene. Now MYC plays a big role in cell division, and the MYC gene is expressed only when it's needed, only when certain cells need to divide. On the other hand, on chromosome 14, there's an immunoglobulin gene. Immunoglobulins are essentially antibodies, and these are essentially expressed constitutively, so they're pretty much expressed all the time. And what can happen sometimes is a translocation event can occur. And as a result, the regulatory region, essentially the promoter, if you want to think of it that way, the promoter region and any other enhancers and so forth, or promoter proximal elements, are located, or I should say become located, right next to the MYC gene. So as a result, the MYC gene ends up being expressed constitutively. As a result, that can trigger uncontrolled cell divisions and essentially lymphoma. Let's now shift gears entirely, and let's talk now about changes that can occur in terms of the number of chromosomes within cells. So on one hand, we can talk about euploidy. Euploidy refers to a variation in the number of complete sets of chromosomes. So in most of our cells we have 2 of each chromosome, so we'd say we'd have 2 sets. Sperm and egg, of course, we just have 1 set. So we can have large-scale changes that can occur regarding the complete sets of chromosomes. In addition, we can talk about changes in individual chromosomes. That will be referred to as aneuploidy. So trisomy 21, for example, we say that's an example of aneuploidy because it's a change in just one of all of our chromosomes. You're all familiar with haploid, diploid, and so forth. Well, it turns out there are situations where various organisms, plants as well as animals, can have extra sets. So we say these are polyploids. So, for example, we can talk about triploids, 3 of each chromosome, tetraploids, 4 of each chromosome, pentaploids, hexaploids, and so forth. And so we just see a couple of illustrations here. For example, the grapes on the left, we have diploid grapes. On the right, we have tetraploid grapes. On the image on the right, we're looking at the leaf of tobacco cells, diploid, tetraploid, and octoploid. Notice that with an increasing number of sets of chromosomes, so an increasingly higher ploidy level, generally that results in larger and larger cells. There's often a correlation between cell size and ploidy number. There is a situation where organisms, or I should say species, can arise by a variety of mechanisms, but one of which is a situation where the genome of an organism essentially becomes doubled. And how this can happen quite often, for example, is an error in meiosis. So instead of producing haploid gametes, sometimes organisms can end up producing diploid gametes. And so if you have 2 diploid gametes fuse, that can result in a tetraploid organism. So as one example, this can happen in animals. It happens in plants quite often, but here we have a diploid frog, namely the Cope's gray tree frog, and on the right is a tetraploid gray tree frog. So it turns out these are 2 different species, but the evidence is that the tetraploid version arose simply as a doubling of the chromosome sets within the Cope's gray tree frog. So in that situation, we say that's an example of an autopolyploid because we had a doubling of an individual organism's or a species, their own chromosome number. Now, on the other hand, we can also form new species referred to as allopolyploids. So wheat is a classic example. So allopolyploids, we're talking about hybridization events between 2 or more different species. And again, often they result in fusion of gametes that have 2 or more sets of chromosomes. So wheat is a classic example. It turns out wheat is a hexaploid. So essentially, the wheat that we have growing in our fields right now came about as a result of essentially 3 different ancestors over time, each of which contributed 2 sets of chromosomes to cells. So as a result, again, wheat is hexaploid. In other words, it has essentially 6 of each chromosome within its cells. And if you think about various organisms or various species with multiple sets of chromosomes, and you think about how these chromosomes line up during meiosis, toward the top here we have kind of your standard diploid situation. So we always talk about meiosis and chromosomes lining up as homologous pairs, and that's what's illustrated here. So we say, well, they line up as bivalents. If you have an autotetraploid, recall that's where you have a genome of an individual organism or species doubling. Those chromosomes line up during meiosis essentially as pairs of pairs. So they line up as quadrivalents, and that's what's illustrated here. If you have something like an allotetraploid, whereby you had hybridization of 2 different species, it turns out they have very similar, or they often can have very similar chromosomes, but they're not exactly homologous chromosomes. We call these homologous chromosomes. So again, these are very, very similar chromosomes that came from separate species that hybridized, and what they tend to do is line up as bivalents, much like we see with a typical diploid situation, and that's illustrated here. And Aneuploidy, recall, refers to the change in the number of individual chromosomes. So a few examples, you've heard of these before, right? Turner syndrome, this is a situation where a person has maybe one X chromosome but lacks an additional sex chromosome. Klinefelter's, that's an XXY situation. Down syndrome, of course, is trisomy 21, so 3 copies of chromosome 21. Recall that often these situations come about as non-disjunction, where we end up with gametes that have either one extra or one fewer chromosomes. So this is a figure that illustrates non-disjunction. So non-disjunction can occur either during meiosis 1, and that's what we see illustrated toward the top. If so, we end up with half of the gametes with an extra chromosome and half of the gametes lacking that particular chromosome. Or it can occur during meiosis 2, and that's illustrated toward the bottom. If that's the situation, again, we can end up with a gamete that has an extra chromosome, a gamete that lacks that chromosome, or we can also end up with gametes that have the normal number of that chromosome. So depending on whether non-disjunction occurs during meiosis 1 or 2, that can result in slightly different outcomes. Just some terms that you might come across regarding aneuploidy. So null isomic, that's a situation whereby for a particular chromosome, a cell is missing both of them. Monosomy or monosomic, that refers to a situation where a cell is just missing one of a pair of chromosomes, Turner syndrome, for example. Trisomy or trisomic, that's a situation whereby cells have an extra chromosome, so trisomy 21, for example. So that concludes our discussion of large-scale chromosome changes.